Assalamualaikum and a very good morning to all students. This is going to be another asynchronous teaching and learning where I'm going to explain and we're going to learn about a new topic of which I've mentioned before, the digestive system. Okay, the digestive system. There's two parts in here. However, for the first part, to share screen. Yeah. All right. So yeah, already share the screen. Yeah, hope you can see it now. So this part will be divided into two parts, which the first one is more on anatomy, and the second one is rather physiology. And the first one, I'm not going to cover extensively since you can read it yourself. Please do read this one, and I'm only going to explain to you the second part of the digestive system chapter, which focus on more on the physiology and the process happen within the digestive system. All right, so let's move on. So these are the expected learning outcome of this chapter. The first one, you must be able to describe how each major class of nutrients means, carbohydrate, protein, and fat, is chemically digested. And you must also be able to name what are the enzymes involved and discuss the functional differences among these enzymes. How do this enzyme act on certain macronutrients and also micronutrients? And the second one of this expected learning outcomes is for you to describe how each type of nutrients is absorbed by the small intestine. Okay, let's go for the first macronutrient, which is the most common one and the only type of I would say macronutrients that are recognized by our body system to produce energy, which is the carbohydrate. Starch which is also a type of carbohydrate, is the most digestible type of carbohydrate, and however, the cellulose is the carbohydrate that cannot be digested. Starch is first digested to oligosaccharides, sub to 8 glucose residues long, and then it will be break into disaccharide. Okay, oligosaccharide is a long chain type of carbohydrate, and then it can be reduced or break down into a more simple one, which is disaccharide. Di means 2. Okay, disaccharide, maltose, and then finally will become glucose. Glucose is a simple form of carbohydrate, the most digested carbohydrate. Okay, the most quickest digested carbohydrate. Okay, and then it will be absorbed by your small intestine. If you, let's say, you consume simply glucose, then it will easily not breaking down. It was straight away going to be absorbed by your muscle. But there's issue with simple glucose consumption, where it can lead to glucose rush. It will not good to some people, especially those who have diabetic, where it's best for them to consume glucose that is slowly digested, which is the long chain, rather than the one that quickly absorbed. Okay, this is very much related to the glycemic index of carbohydrate, which I will explain later. Okay. So the process of carbohydrate digestion will begin within the mouth, where the salivary amylase hydrolyze starch into the oligosaccharide. Okay, it will break down starch into oligosaccharide. Okay, and then amylase itself, okay, the enzyme that breaks starch, will work at best within the range pH of 6.8 to 7, slightly acidic, slightly, not too much, just slightly acidic, of oral cavity. Okay, I would say neutral, yeah, just 0.2 below from the normal pH level. Amylase quickly denatured on contact with stomach acid and digested by the pepsin. So amylase quickly denatured will break down and contact with stomach acid and digested by pepsin. So these are the where salivary amylase salivary amylase is produced by your saliva glands. Okay, the submandibular, there's three part of salivary gland within your oral cavity. Okay, the parotid gland, submandibular gland, and sublingual gland. Right, these are the salivary glands that produce salivary amylase. But salivary amylase will stop working in stomach when the pH, the environment pH, is less than 4.5. Okay, gastric acid, hydrochloric acid within the stomach, your gaster. Okay, it's a very strong acid. Okay, the environment, and amylase will stop working there. And 15% of dietary starch digested before it reaches small intestine. So, the breakdown of starch already happened within the oral cavity. Okay, to oligosaccharide. Then when it reaches small intestine, pancreatic amylase converts starch to oligosaccharides and maltose within 10 minutes. Okay, so it's rather quick when, oligosaccharide or, the digested carbohydrate, when it reaches small intestine, okay, pancreatic amylase converts starch to oligosaccharide saccharide and maltose within 10 minutes. So oligosaccharides and maltose contact brush border enzyme. Contact with brush border enzyme. Okay, dextrinase, glucomylase, maltase, sucrose, and lactate, and act upon oligosaccharide, maltose, sucrose, lactose, and fructose into glucose. Then lactose become indigestible after age 4 in most humans due to decline in lactose production. So yeah, when we get older, the baby get older, and after age of 4, changes in our body happens where the production of lactase reduce. So lactase enzyme is responsible to break down lactose. So when it not properly break down, when lactose is not properly break down, okay, lactose is the a carbohydrate from, usually you can find it in the milk, and lack of lactase production, the enzyme responsible to break down lactose. So you will experience stomach upset, unpleasant in your stomach, because it's not the lactose itself not properly digested and break down, and what we call it as lactose tolerance. Okay, so yeah, usually babies produce sufficient amount of lactase enzyme. Okay, and once the persons get older, okay, 4 months and above, the production of lactase become lesser and lesser. So the next one is protein. Protein is the simplest form of protein, that what we call is an amino acid. Absorbed by the small intestine, comes from 3 sources. Okay, the amino acid come from these 3 sources, which are dietary protein, digestive enzyme, okay, digested by each other of the enzyme. Okay, the enzyme itself is a form of protein. And then, of course, the third one is the protein of the body within our body itself. Okay, it's not the epithelial cells digested by the enzyme. You know, in a way, we eat ourselves. Okay, in other words, we actually eat ourselves. Okay? The epithelial cells within the digestive tract. Okay, when it's detached from the lining, of the epithelial cells detached from the lining, it can be digested by the enzyme as well, similar like other proteins that we consume in our diet. So endogenous amino acids from last 2 sources is about 30 grams per day. Okay, that's what is recommended to consume. The exogenous amino acids from our diet total about 44 to 60 grams per day. Okay, the endogenous amino acid source of protein, this is the last 2, the digestive enzyme digested by each other. Okay, the enzyme itself is a form of protein, as I mentioned, and the epithelial cells within the digestive system is also consumed and absorbed by the body. So that's meant by the endogenous amino acid. Okay, so it constitutes of 30 grams per day, and while the exogenous, mean out from the body, type of amino acid, which is our diet, comes from our diet that we consume, must constitute at least between 44 to 60 grams per day. And then okay, peptidase. Okay, peptidase is an enzyme that digests protein. It is an enzyme that responsible to digest protein. Okay, peptidase begin their work in the stomach at an optimal pH of between 1.5 to 3.5. It can only be break down within the very acidic environment, which is the gaster acid within your stomach. Okay, therefore it will not digest it within your mouth. It will only start digest and break down in your stomach when exposed to hydrochloric acid. And unlike carbohydrate, which can easily absorb, so that's why some scientists refer protein as an expensive protein, because it requires a lot of energy. Okay, energy to break it down to produce more energy. Okay, I'm going to explain this later. And then pepsin, hydrolyzed bipeptide bond between tyrosine and phenylalanine. Okay, pepsin digested 10% to 15% of dietary protein into shorter peptides and some free amino acid. Okay, this is another enzyme that is responsible to digest and to break down the protein, pepsin. We continue to digest protein in small intestine. Pepsin inactive. Pepsin will become inactive when it passes through the duodenum. Okay, if you read first the part 1 digestive system slides, okay, the duodenum is part of your small intestine. Okay, and pepsin will be inactivate when it's passed into the duodenum. Okay, and mix with the alkaline pancreatic juice. Okay. Okay, so pepsin, the enzyme that breaks down protein to amino acid, will stop at duodenum. Duodenum is the first part, or the part where sphincter of the gastric, the stomach, okay, sphincter of the stomach connect with the beginning of the small intestine. I'll show you. Yeah. So this is duodenum. Duodenum is the beginning of small intestine. So when hydrochloric acid reacts to protein and peptide is released within this organ, okay, it will break down protein into amino acid, and when exposed, when the food, digested food, continue to digest and pass through the duodenum, okay, the part where the small intestine begin, and the pH will increase a bit, no longer acidic, and that will make the enzyme stop working and stop digesting, stop breaking down the protein at the duodenum stage, right? Yeah. So when it reached duodenum, as I mentioned, the pH increased into more alkaline due to the pancreatic juice. Okay, pancreatic juice. Okay, pancreatic juice is more alkali compared to the gastric juice, which is very acidic. So when it reached to level of 8, okay, slightly alkali, it will stop the production, it will stop breaking down the protein. And pancreatic enzyme, which are trypsin and chymotrypsin, okay, will take over the process. Okay, will take over the process of breaking down protein. So hydrolyzing polypeptides into 7 shorter oligopeptides. It will hydrolyze, the protein that has been break down within the stomach into smaller protein, which is polypeptides. Okay, from polypeptides into oligopeptides. Poly means if you notice the prefix here mentioned, it's poly means multiple. Oligo means, this one certainly. Okay, oligo means 8, if I'm not mistaken. Few. Okay, prefix of oligo means few. Yeah, oligo is octa is 8, right? Oligo still multiple, but I presume it's shorter than poly. Poly versus oligo versus poly. All right, let's see. So the prefix attached to the word such as oligo means few, whilst poly means plentiful. Okay, so in other words, oligo is lesser than poly. It's still, no long chain type of amino acid, but oligo is shorter than poly. Poly is more longer compared to oligo. Yeah, so that's that. So it will hydrolyze polypeptides into even shorter oligopeptides. Okay, so oligopeptides, when exposed to alkali environment from the pancreatic juice, oligopeptides taken part one amino acid at a time by three more enzymes. Carboxypeptidase, aminopeptidase, and dipeptidase. Okay, these are the enzymes that are responsible to further break down oligopeptides. So it will remove the amino acid from the COOH at the end of the chain. Okay, so that's the function of carboxypeptidase. Okay, we remove OOH from the amino acid, and aminopeptidase will remove them from the amino ends. Okay, whilst dipeptidase will split peptidase in the middle and release two free amino acid. Okay, if you can see oligopeptidase, I will show you. Oligopeptide. Okay. So oligopeptidase will taken apart, will break down one amino acid at a time by three more enzymes. So it will break down by these three enzymes. Okay, these three enzymes is responsible to further break down the oligopeptides. Oligopeptide image. Okay, this is COOH I mentioned just now that only can be detached by carboxypeptidase, COH, okay, when um, um, when oligopeptide exposed to carboxypeptidase that is within the pancreatic juice, okay, that available in pancreatic juice, all these three peptidase enzymes in the pancreatic juice, okay, it will detach COOH, okay, and then aminopeptidase remove them from the amine, okay, H2, okay, and H2, this one, okay, aminopeptidase will remove this one, okay, we'll remove this, we'll remove this chain, okay, and the peptidase split peptides in the middle and release two free amino acid. Okay. So NH is the amino acid. This is carboxyl group. The amino group. Okay, NH. Let's see. So this is amino acid. Okay, it must have N, C, and O. Okay, it like CHO but with nitrogen. That is basically the component that constitute amino acid. Okay, it seems like carbohydrate but with nitrogen. So yeah. So yeah, dipeptidase split peptidase in the middle. Okay, split peptidase in the middle. Okay, right, peptidase, split peptides in the middle, dipeptides. Sorry, dipeptides in the middle. Dipeptides in the middle. This one. Dipeptides in the middle and release two molecules of free amino acids. That's how it's being break down. It will break down the protein into amino acids. So it happens within the duodenum. Duodenum, of course, within the gaster, to break down the polypeptides to oligopeptides, and then oligopeptides will further break down at the duodenum, at the duodenum part, and becomes amino acid. Okay. Yeah. So this is the pancreatic enzyme takes over protein digestion in the small intestine, which is duodenum, the beginning of small intestine after the sphincter. What's this? Okay, by hydrolyzing polypeptides into shorter oligopeptides. Yeah. So the breakdown of protein will happen in the gaster. Will make it break down to oligopeptides, and then oligopeptides to protein to polypeptides, and then it will bring the more simpler versions of protein or peptides to the small intestine, the beginning of small intestine, which is the duodenum, and then within this area, pancreas juice will flood in, and then change or break down the polypeptides to oligopeptides and other three type of enzyme, peptidase, dipeptidase and aminopeptidase will further break down the oligopeptides to produce two amino acid. It is what happened. This is what happened. Okay. So this is pancreas. This is duodenum, your small intestine. Okay. So in this picture, it's stretch the duodenum to make it straighten. Then yeah, this is what the process happened. So polypeptides will become oligopeptides, and then these are the response of carboxypeptidase. And then yeah, and the next type of macronutrient is lipid, which is fat. Okay, hydrophobic. It is hydrophobic, meaning afraid of water. Its hydrophobic quality of lipids makes their digestion and absorption more complicated compared to the other first two type of macronutrient. Lipases is the enzyme responsible for fat breakdown. So there's a lingual lipase secreted in the salivary gland. Okay, apart from the amylase enzyme that breaks down carbohydrate, there's also lipases in your saliva. So it's active in mouth, but more active in stomach along the gastric lipase. So another way, since carbohydrate and also fat shares the same constitution of which of consists of carbohydrate, carbon, oxygen, and hydrogen. So it speaks volume that these kinds of macronutrient will be absorbed or start to digest it within the oral cavity, since it shares the same how to say, same um, what do they call it, same type of atom but different shape, molecule shape. Yeah, you get what I mean. Then yeah, it will be more active in gastric lipase. So gastric lipase is also available within the stomach, gastric means stomach. And then 10 to 15% of lipids digested before reaching duodenum. So yeah, part of lipid, not too much. Okay, at least 10% and maximum of 50% of lipid that you consume only be digested within your stomach before it reaches the duodenum. Okay, unlike protein, okay, most of them already been digested at the duodenum stage. So pancreatic lipase is small intestine digest most of fat. So pancreatic lipase that produced by the pancreas. Okay, apart from enzyme peptidase to break down protein. Pancreas also produces enzyme that break down lipid. Okay, and pancreatic lipase in small intestine digest most of the fat. Okay, break down most of the fat. And fat enters duodenum in large globules exposed to lipase only at the surface. Okay, and globules broken up into smaller emulsification droplets by certain components of bile, lecithin, and bile acid. The bile that break down all the globules, okay, globules that earlier has been break down by the pancreatic lipase. So agitation by the segmentation breaks of the fat into droplets as small as 1 micrometer in diameter. So it becomes smaller and smaller droplets. Fat droplets will be break down to smaller size. Okay, as small as 1 micrometer in diameter. The coating of lecithin and bile acid keeps it broken up. Okay, exposing far from surface to enzymatic action. Okay, so that's that. Okay, there is enough pancreatic lipase in small intestine after a meal to digest the average daily fat intake as little as 1 to 2 minutes. Okay. So lipase act on triglyceride. Okay, like triglyceride is the storage form of lipid that circulates within our body. It constitutes of 3 molecules of fat, 3 molecules of free fatty acid. The triglycerides. Okay, type of fat, type of lipids found in our blood. Okay, hang on. So this is triglyceride, constitutes of glycerol and 3 free fatty acids. Okay, one molecule of glycerol with 3 free fatty acids, that's triglyceride. So yeah, lipase can break down triglyceride, where it will detach glycerol from the molecule of triglyceride and form 3 molecules of free fatty acids. Three fatty acids, which constitutes of 2 free fatty acids and 1 monoglyceride. So glycerol is the backbone of the triglyceride. So we remove it and leave the middle one. So the product of lipase action were 2 free fatty acids and also monoglyceride. Monoglyceride is used to be a free fatty acid as well, as I shown here. Okay, 3. But free fatty acids, lipase itself will react on the third free fatty acids to produce monoglyceride. Okay, so this is a fat digestion and absorption. Okay, this is what fat globules that I mentioned just now. It will break down to smaller compartment. Lipase will do that. Okay, lipase, the enzyme. Okay, pancreatic lipase. It will break down the globules into small globules, to smaller droplets. Okay, smaller droplets, okay, which is lecithin and bile acid. So become this emulsification droplets. Fat globules is broken up and coated by the lecithin and bile acids. Okay, lecithin and bile acids. Okay, lecithin and bile acids will combine, emulsificate with the smaller fat globules. Okay, you so bile acid have 2 regions, the hydrophobic and hydrophilic, and the one that hydrophobic will attach the fat, since it makes sense. It's friendly to oil things, whilst the hydrophilic one will expose to the cytoplasm or water environment. And the absorption of free fatty acids, monoglycerides, and other lipids depend on minute droplets in the bowel called micelles. Okay, absorption of free fatty acids, monoglycerides, and other lipids. Okay, if you remember, free fatty acids from the triglyceride and also monoglyceride, and then from the third free fatty acids that we break down by lipase. This kind of droplets, what we call it as micelles. It made in the liver. It consists of 20 to 40 bile acid molecules aggregated with the hydrophilic side groups facing outwards and their hydrophobic steroids ring facing inward. Hydrophobic. Okay, the bile acid. Okay, this is produced by the liver. So bile, phospholipids, and cholesterol diffuse into the center of the micelles to form its core. Okay, and then micelles pass down to the bile duct into the duodenum. Okay, with the droplets that combine with the acid biles and also lecithin foci. So yeah, my cells pass down the bile duct into the duodenum, okay, where they absorb, okay, fat soluble vitamin. Is fat soluble vitamin like vitamin A, D, E, K, okay, all vitamins except B and C, those are water soluble, so it will absorb fat soluble. My cells, okay, pass down the bile duct to the duodenum, and then will absorb all the fat soluble vitamin. More cholesterol will absorb also more cholesterol, and the free fatty acids, and also monoglycerides produced by fat digestion. This is the functions of the bile duct in the duodenum. It absorbs, okay, more cholesterol. They transport lipids, okay, and then they can transport lipids to the surface of the intestinal absorptive cell, okay, within the small intestine, and lipid leave the micelles and diffuse through the plasma membrane into the cells. Okay, micelles are reused. Okay, micelles can be reused, packing up other lipid to cargo and other lipids, transport them to the absorptive cells, which is the wall of the duodenum, the wall of the small intestine. So fat hydrolysis, this is the dietary lipid. The fat and lecithin will attach to the droplets, and also bile acid as well. This is triglyceride, okay, triglyceride, and glycerol. This is glycerol. So yeah, pancreatic lipase, it will react. Pancreatic lipase is the enzyme. When it react to the triglyceride, it will produce 3 free fatty acids and 1 monoglyceride. Okay, glycerol with one chain of free fatty acid. So monoglyceride and also free fatty acids. These true fatty acids break down by the peptides, pancreatic lipase. So yeah. So this, together with other cholesterol and also fat soluble vitamins. Okay, this is fat soluble vitamin, also cholesterol. Okay. Okay, we form a lipid core. Okay, is this in this lipid core in this particular bile? It will bind with the bile acid. Okay, bile acid will bind with them. So micelles in the bile pass the small intestine and pick up several types of dietary and semi-digested lipid. Okay, this is semi-digested lipids. And also other dietary lipids. Yeah. So that's all about the brief explanation of 3 macronutrients. And then let's look into the micronutrients, micro vitamins and mineral. So vitamins absorbs and change. So fat soluble vitamins, I mentioned, is A, D, E, K, absorbs with the lipids. Lipids, if they are ingested without fat containing food, they are not absorbed at all, but are passed in the feces and wasted. And water soluble vitamin B complex and C absorbs by simple diffusion. Okay, just using simple diffusion, it doesn't have to be further digested. And B12, if it's B12, it's bound to intrinsic factor from the stomach. Okay, so normally B12 is the vitamin that assists digestion. Okay. So yeah, other part is other type of micronutrients is the mineral, mostly in the form of electrolytes. So it will absorb all along small intestine without any, breakdown requirement. So sodium co-transports with sugar, it will co-transport with carbohydrate and amino acid. Chlorine exchange from bicarbonate, reversing chloride bicarbonate exchange that occur in the stomach. That's chlorine and sodium. Whilst iron and calcium absorb as needed in your dietary, iron absorption is stimulated by the liver hormone. The liver hormone will help to absorb iron. Okay, and the name of the hormone is hepcidin. So it will absorb cell spine diffuse iron, Fe2+, and internalize by active transportation. It requires ATP to absorb iron, and that will be responsible for hepcidin hormones. Okay, to absorb iron in the digestive system, particularly within the small intestine. Okay, unable to absorb ferric iron. Okay, it unable to absorb Fe3+. It can absorb ferrous, but it cannot absorb ferric iron. But the stomach acid will reduce ferric iron. Okay, it will reduce ferric iron, so it can become ferrous and then absorbed by the body. Okay, so the stomach acid will change ferric to ferrous before it can be absorbed. So transferring the extracellular protein transport iron into the blood to bone marrow, muscle, and liver. So transferrin will transfer the iron to bone marrow, muscles, and liver to produce blood. Basically, usually iron is responsible to produce blood production. And then transferrin, the enzyme, that is the type of enzyme responsible to bring all the ferrous into bone marrow, muscles, and liver. So calcium is absorbed throughout the intestine by different mechanism. Calcium, calcium or potassium. Okay, it uses active transports to the duodenum. So we will enter through the calcium channel. If you remember action potential, so it requires some ATP to be absorbed into the system and in apical cell membrane and binds to calbindin protein to concentration gradient will continue to favor calcium influx. So it will continue this active transportation in the duodenum to absorb calcium. Potassium will actively transport out of base of cells into bloodstream by calcium ATPase and sodium potassium antipode. Okay. And the absorption also occur with the diffusion between epithelial cells in jejunum and ileum. Okay, jejunum is a part of small intestine after duodenum, and ileum is the last part of small intestine. So the long small intestine can be broken up into three parts: the duodenum, jejunum, and also ileum. Okay, ileum is the name it gives because it's near to iliac bone. Okay, the hip bone, iliac bone. If you remember, iliac region. So yeah, the end part of the small intestine is located at within, nearby the iliac bone. That's why it names ileum. So yeah, other type of minerals that we absorb are types of hormones. Okay, apart from enzyme, hormones also responsible for digestion. Parathyroid hormone. Parathyroid hormone is located, near your throat here. Parathyroid hormones secreted in response to drop in blood calcium level. Okay, if your body notice that drop in calcium level within your body, so parathyroid hormone will secrete, and then this will ask, will ask the to stimulate kidney to synthesize more vitamin D from the precursor made by the epidermis and the liver to produce more calcium. And then that particular vitamin D synthesized from the precursor as the precursor will be absorbed in the cell in the jejunum in 3 ways. And then parathyroid hormone increase the level of calcium in the blood. Okay. So vitamin D is the precursor, affects an absorptive cell of the jejunum in 3 ways. Okay, will increase the calcium by 3 ways, which are increased number of calcium channel in apical membrane, and then the next one is increase the amount of calbindin in the cytoplasm to produce more calcium, and then increases the number of calcium ATPase pump at the basal membrane. So there's 3 pathways that vitamin D will take part in increasing back the drop or reduced level of calcium in the body. And then of course water is a type of nutrients that are oftenly forgotten. Usually people will always mention about macro and micronutrients, but forget about water. Water is also important in our digestion. So the digestive tract receive about 9 liters of water a day. And 0.7 liters in food, 1.6 liter from the drink, and 6.7 liter from the gastrointestinal secretion, okay, from the activity of digestion. Quite huge amount, okay, of water can be produced by the chemical reaction within the GI tract. So 8 liters is absorbed by small intestine and 0.8 liter by large intestine. So most water will be absorbed in your small intestine, and just 0.8 liter, less than 10%, okay, is absorbed within your large intestine. Sorry, less than 1%. Less than 1%. Less than 10%. Okay, and 0.2 liter voided in daily fecal output. So some of the water content will secreted, okay, excreted through the feces. Okay, if not, if your feces is too dry, it will injure your sphincter. Yeah. And water is absorbed by osmosis. Okay, the chemical reaction of osmosis. Water is absorbed that way in our small intestine and large intestine, following the absorption salts and organic nutrients. They absorb together, okay, with other organic nutrients and also salt. So diarrhea occurs when large intestine absorb to little water. Okay. So feces pass through too quickly if intestine is irritated. When the intestine is irritated, it doesn't absorb water efficiently because kind of inflame of your large and small intestine, and makes the feces contains high concentration of solutes, lactose. So when you have lactose intolerance and you eat something with high lactose, you will have diarrhea. Okay. Constipation occurs when the fecal movement is slow. Okay. Tidak lawas lah. Constipation is tak lawas. Yeah. Too much water gets reabsorbed, and your feces become hard, and it's difficult for you to secrete the feces. Excrete the feces. Say the word. Excretion. Yeah, excrete. Okay, excrete the feces. Feces means your waste lah. So large intestine receive about 50 milliliter of indigestible residue per day, and it eliminates feces by defecate. Okay, excretion, defecate. Okay, eliminate the feces by defecation. The process of get rid of the feces. Okay, question. Large intestine measures about this is the size of large intestine. Okay, measures about 1.5 meter long and 6.5 centimeter in diameter, and begins as cecum inferior to ileocecal valve. Okay, it begins there. Cecum inferior, ileocecal valve. The ileocecal valve is the valve. Ileocecal valve is the sphincter muscles situated at the junction of ileum, the small intestine, to the large intestine. So ileocecal valve. We start from there. So that's the ileocecal valve. Your ileum, small intestine, you can see it. Okay, cecum. We start from there. Cecum and ileocecal valve. This is appendix, and this is the ascending colon of your large intestine. So you start from there. Begins in the cecum inferior, and also cecal valve, ileocecal valve. Okay, ileocecal. Ileo means ileum. Cecal means this part, cecal here. So ileocecal valve. Yeah. We got how the words comes from. So appendix. Okay, the appendix attached to lower of the cecum, densely populated with lymphocytes, and it's a significant source of immune cells. So basically, some people say that appendix doesn't really have any function, but actually it's a house of a lymphocyte, which acts as the defensive mechanism from microorganism. Okay, because starting from here, microorganism is already developed. Okay, all the gut flora is already developed in here. So lymphocyte will act as the defensive mechanism to avoid any infection happen within the during digestion process. And ascending the first part of large intestine that go upwards. Okay, right colic, hepatic. Okay, flexure, transverse colon. Transverse is the one that yang melintang tu. Left colic. Okay, splenic, flexure, and also descending colon. Okay, large intestine. Yeah. Ascending, transverse. Here you can see it. Ascending, transverse, and also descending colon. Sigmoid colon. Sigmoid is it curves a bit, and then rectum, and then anal, sphincter. So sigmoid colon is the S-shaped portion leading down to the pelvis. Okay, this is the picture. Actually, I do have a picture here. So this is the anatomy of a large intestine. Yeah. Cecum, ascending colon, descending, transverse colon, descending colon, sigmoid colon, rectum, and then sphincter, anal sphincter, yeah. So yeah. So rectum is the portion ending at the canal, the anal canal, has 3 curves and 3 infolding called the transverse rectal fold, rectal valve, it has 3 folds and 3 foldings, and then anal canal, the final 3 centimeters of large intestine passes through levator any type of muscles, the name of the muscles, and pelvic floor terminates at the anus, terminates out from the anus, and anal columns and sinuses exudes mucus and lubricants into anal canal during defecation. Large hemorrhoidal veins for superficial plexus in anal column and around orifice. Hemorrhoids is a permanent distended veins that protrude into the anal canal or form bulge accessible to the anus. Hemorrhoidal. Muscularis externa of colon is unusual, so it's not really important. You can read this, okay? Taenia coli, a longitudinal fiber concentrated in the 3 thickened ribbon-like strips, pustra pouch in the colon caused by the muscle tone of the Taenia coli, Taenia coli. Internal anal sphincter, smooth muscles of the muscularis externa. An external anal sphincter is the skeletal muscle of the pelvic diaphragm. An omental, epiploic appendages, acute-blood fatty pouches of peritoneum adhering to the colon. Omental. Yeah, this omental appendages, like a bile, bile duct. Function is still unknown. So yeah, And then the last part of our digestive system is defecation. Defecation. Where it starts? Okay, this. Okay, stretching of the rectum stimulates defecation reflexes. Okay, stretching when there is a feces go down from the rectum, it will stretch the rectum, stimulate defecation reflex, and then accounts for urge to defecate. Okay, keinginan untuk defecate that is often felt soon after a meal. The intrinsic defecation reflex works entirely within the myenteric plexus. Stretch signals travels through the plexus to muscularis, causing it to contract and the internal sphincter to relax. So it's relatively, where is it? Relatively weak contraction. So, and then the final part, okay, parasympathetic defecation reflex. This is, parasympathetic involves in spinal cord. Involves spinal cord, stretching of the rectum, sensory signal to spinal cord, and pelvic nerves return signal, intensifying the peristalsis, get the movement to defecate, and relaxing the internal of the sphincter, and defecation occur. Okay, this is the process, the mechanical process of defecation. So abdominal contraction. Okay, abdominal contraction when you defecate, the process of defecation is what we call this as Valsalva maneuver. The Valsalva maneuver, menekan, increase abdominal pressure as a levator any lifts anal canal upwards. So therefore, feces will fall away. Okay, for that, thank you so much. That's the topic of the digestive system. So thank you.